Our next presentation is Orca Bio, and I'd like to welcome Dan Kirby, Chief Commercial Officer, to the stage.
Hi, everybody. Love the 5 o'clock time slot, so hopefully I'm not between you and a happy hour. I'm Dan Kirby with Orca Bio. Orca Bio is a cell therapy company based in the Bay Area that spun out of Stanford in 2016. There's our disclaimer and a little about our company. We have 2 products in trial right now. We have Orca T, which is in a phase 3 trial randomized against standard of care allo transplant to show superiority for chronic graft versus free survival as well as overall survival. Orca Q, our second product, is in clinic in single-arm trials right now, advancing into hemalignant non-perfect match donors as well as in the autoimmune space. We have expanded our focus from general hemalignancy and AML, ALL, and MDS, which Orca T is focused on, into other malignancies and combining with CAR-Ts. We also are pursuing expanding the market into autoimmune, starting with a PPMS trial, primary progressive multiple sclerosis with Orca Q. We have a clear path to commercialization when our data reads out, which should be early next year. If you look at the fundamental science of Orca, what we're doing is replacing the immune system. And that's not new. That's happened with the standard allo transplant for 40 or 50 years. And it's been shown to work. You can see here in the bars, both in oncology and non-oncology indications, just exactly how much evidence there is in the publication, incidence and publication of replacing the immune system for these patients. We know proof of concept works here, but the problem is toxicity. Allo transplant is a very toxic approach to therapy. It works, it's a patient's chance for a cure, but we can do better. As a healthcare community, and our physicians are very much aligned with that. 40% of the patients die within the first 3 years, and they die either due to primary relapse or due to, whether it's infection, GVHD, et cetera, complications. Some of those complications directly related to the intervention. At Orca Bio, our platform is a one-to-one matched donor-to-patient approach. So allogeneic, yes, but not allogeneic off the shelf. We rely very much on matched donors and semi-matched donors based on our program. Our platform actually was developed at Irv Weissman's lab at Stanford, and Irv mapped the human stem cells, I'm sure a lot of you know, and that's where the company spun out of. Essentially, our platform is able to identify and isolate any subpopulation of cells to create and custom build a new immune system in a patient. Our lead asset, Orca T, the one in the phase 3 trial that's completed, is highlighted right here. We take day 0 from a donor, stem cells, and highly purified Tregs, and then we allow those 2 days to proliferate inside the body, all of our expansions in vivo, not ex vivo, and then 2 days later we put in a previously cryoed amount of T-cons that go in and destroy any residual disease in that patient, but because the Tregs have been purified and have proliferated inside the patient, they do not attack the organ or the other tissues as much as a standard allo would. And we'll go through data in a minute. Our pipeline starts in the hemalignant space, where transplant's already established a market. And here you can see with Orca T, we are in AML, MDS, and ALL. Now we will be, upon approval, success of the trial, the first cell therapy approved for MDS and AML. As we look at the other indications, we've treated with haplo donors with Orca Q, again, non-matched donors, or semi-matched donors, I should say, and we've also experimented using with CAR-T. So again, a lot of other applications with our platform and our products with it. I mentioned multiple sclerosis. We have cleared IND and we'll be starting patients, hopefully by the end of this year, on a trial looking at primary progressive multiple sclerosis with Orca Q. If we go back to the hemalignant space in Orca T and the baseline indication of AML, ALL, and MDS, you can see about 31,000 patients are diagnosed each year with these indications. Yet only a small fraction, about a third of them, make it to a transplant center and receive a transplant. We talked about the reasons why, but also, too, there's a tremendous opportunity once you have a better intervention to be able to address some of that unmet need in the market. We did additional work with transplanters and really asked, of the patients that go there, are they all receiving? They make it to your center, but are they receiving therapy? And what we got was, on average, in our indications, about 20% of them never receive therapy. They're thought to be too frail for standard of care. And we believe ORCA-T has potential here. Now, this is a survey of physicians that we've done of transplanters inside the US. And you can see on the bar, their growing appreciation for trade-offs that exist, whether it's a graft versus host disease versus graft versus tumor, meaning that if you decrease the toxicity, you'll also decrease the efficacy. And then also, too, with other means of GVHD intervention, you actually could trade GVHD for infection. And really where the receptivity goes, and these are the numbers on the right-hand side of the slide, 3/4 of the physicians are receptive to using an engineered graft or a third-party approach to put a cell therapy in. 80% agree with the promise that purified cell therapy is a key to unlock the improvement in patients. 82% agree that this approach will select cells the most likely to control the disease without putting cells in that'll control or expand the toxicities. ORCA-T data, and again, these are patients from our single-arm trials that were eligible for our phase 3 study compared to a contextual cohort. Our phase 3 is randomized, so we will have head-to-head data against the standard allo upon data release. But looking at this subset of patients, and this is using 86 patients in standard of care and 57 patients that were treated with ORCA-T with our phase 3 regimen, you can see our primary endpoint, chronic GVHD-free survival, significant improvement over standard of care. Overall survival, relapse-free survival, and GVHD and relapse-free survival showed similar results. And I will give a little bit of a caveat that the contextual cohort we used was Stanford here, which Stanford has traditionally higher responses, or better responses, I should say, than what the CIBMTR registry has. So again, against a very strict cohort contextually, we perform very well with our single-arm patients, and we have treated in this subset 57 there. I'll talk about the phase 3 trial in a few slides. As we actually go to what this profile could be, we tested a base case and an upside case of our profile with US physicians, and we received very positive results from them on where they would use ORCA-T and what the potential market penetration could be in the existing market. This is not expansion, this is the existing market, where we can hit 2/3 or almost 3/4 over to ORCA-T with our profile. Our phase 3 trial, I mentioned before, it's a randomized trial against the standard of care allo. Our primary endpoint is that chronic GVHD-free survival that I talked about. We participated in 22 centers in the US, 22 of the top centers, so a lot of enthusiasm from the treatment centers to being involved in this trial and using ORCA-T. And then, of course, it was 174 patients, which are randomized 1-to-1, ORCA-T versus standard of care. We have passed the number of events that we would need to read the trial out. They're being adjudicated now. We anticipate data to be released in the first part of next year. We have RMAT designation. We've had discussions with both FDA and EMA. We will move forward to file in the US shortly after data. Our manufacturing facility is built and will be operational with product this year as we finish our trial and go into our registrational phases, into our approval phases with the FDA, pending data. But we can actually supply the US market out of this plant, and we're also looking East Coast and European locations as we look to expand the patient population, not only for ORCA-T, but ORCA-Q and future products. So looking at where the opportunity is, I talked a lot about heme malignancies, and with our platform, we can address in AML, ALL, and MDS a significant number of patients. But that's not where we want to stop with ORCA. That's just our beginning. As we get to other blood cancers, multiple myeloma, lymphoma, whether it's by ourselves or in combination with CAR-Ts, which data has already been released of ORCA-T with CAR-T, and you'll see more probably later this year. We can see other blood cancers can almost double the market. As we get into select autoimmune, that's really where the potential can move forward. And again, with this, we start with primary progressive multiple sclerosis. We keep working with our products with it, but the idea of replacing an immune system in an autoimmune patient has tremendous potential. And as always, we want to end on a patient. This is Mikhail. Mikhail was 71 years old, was actually treated at one of our trial centers with Orca T. He was at first told no, he wasn't going to be able to receive therapy, whether it's standard allo or an Orca T. They actually consulted with another center on the other side of the country, and between the two centers, they made the decision to move forward. Our protocol does allow over 65, so he was eligible for our phase 1B trial. Mikhail was actually profiled in Wall Street Journal before JPM of 2023, and he received an Orca T. He is about 4 years out from therapy. He did not receive any GVHD, so again, he didn't have the toxicities a lot of these patients suffer, and he's alive and doing well to this day. So with that, I want to thank you all for your time and attention, and I think I'm a little early, so I can hand over the next speaker and hopefully get you back on track. Thank you very much.